Here we are at our final discussion about epithelial tissue. This is our discussion of glandular epithelium. This is a type of epithelium that's going to be specialized in somewhat. It's specialized in the sense of being able to produce and secrete material into a duct or into an environment. And so as we look at our epithelium that is specialized for secretion, we call it glandular epithelium. So we're first going to look at the different types of secretory modes in which cells can produce the material. We're going to define the different ways these molecules can signal cells, either local cells or cells at a distance. We are then going to be able to classify glands. We're going to classify them on what their secretory product might be, the structure of the glandular epithelium, and also the structure of the ductal system that transports that material from the gland to its target cell. And lastly, we're going to be able to compare and contrast mucous versus serous glands and know the similarities and differences between those. Those are going to be our predominant type of cell that we're going to be looking at. So, glandular epithelium is going to be that epithelium that is surrounded by connective tissue and produces a secretory product in a structure called a gland. Now, the epithelium that actually produces the material is referred to as the parenchyma of the gland. And then we have other tissues surrounding that epithelium, and we simply refer to that collectively as the stroma of the gland. Now, in this histological slide, what we're looking at principally is going to be our glandular epithelium. This is the parenchyma that we see. It's going to be a nice rounded structure, and we're going to classify these as an acinus. Very center, this is going to be our free surface, that little small circle. That's the lumen of our glandular epithelium. And you can see some of the cell boundaries. We've got our nuclei. So these are going to be nice pyramidal shaped cells. And again, if we had to classify these, we would say this is going to be simple cuboidal because of how high and wide they are, even though they're pyramidal shape. Some of the material in these is going to be slightly different. You can see the nice reddish stain to these cells versus this different color. And that's going to help us identify secretory material. The tissue surrounding the parenchyma that we might see here, this is part of the stroma. And although your visual field immediately goes to this nice, big, broad luminal area here, here, and here, we will very quickly be able to identify these as blood vessels with cells in them. We have a few cells in here, but mostly an empty blood vessel. And this is going to be one of our ducts for our glands. The SD stands for secretory duct, and I believe this is going to be 2 layers of nuclei. You can see these sort of in the background. 2 layers of nuclei, and this might be a stratified cuboidal epithelium. So a nice big duct. This is not part of the parenchyma. This is not producing any secretion. It's transporting that secretion. Here we have the parenchyma producing the material. Now, the product from a particular gland, the chemical signalers are referred to as cytokines. And these signalers can act in a number of different ways. These signals often can feed back directly to the parenchyma, the gland that is producing the material, in which we call that an autocrine type of cytokine. Or the cytokine can react locally to other cells in the vicinity, and this is called a paracrine type of cytokine in a secretory mechanism. It doesn't enter into the bloodstream, but does in fact act locally. The last is the endocrine type, and you've probably heard of endocrine glands, and these are certainly ones that will react at a distance. They're targets at a distance, and they travel to that target cell through the bloodstream. So endocrine cytokines are going to be transported in the bloodstream and affect tissues at a distance. It's not that they can't react locally. They can do that also, but they're principally going to target cells at a distance based on which cells have the receptor for that particular cytokine. In this illustration, we can simply see autocrine mechanism in which we have a cell producing a particular cytokine, but the cell also is responding to a stimulus by producing a receptor as well and displaying it on its cell surface. These cytokines then feed back, bind to the receptor, and affect the activity of this particular cell. This is autocrine in its secretory mechanism, producing the secretion, the cytokine, and responding to that through its receptor. Now, as we look at this particular example, we are looking at two different cytokines. This is transforming growth factor beta, but we also have platelet-derived growth factor, and anytime you see a R here, that's going to be a receptor. So the green receptors you see are for platelet-derived growth factor, and the blue is going to be responding, these are the receptors for transforming growth factor beta. So in this case, if you have a small amount of transforming growth factor beta, that will bind to the receptors, that will signal the release of platelet-derived growth factor antigen, and that binds back to its receptors, and it leads to a proliferative event. However, if you have an abundance of TGF-beta, it will bind to the receptor. You have this platelet-derived growth factor. It's being produced. That binds back to the receptor, but notice what happens. With an abundance of transforming growth factor beta, you have a downregulation of the PDGF receptors. Here we see 5 at sort of resting conditions, but in response to a huge amount of TGF-beta, now we're down to 1. We've downregulated those. So even though the amount of the cytokine, the PDGF, is the same, this autocrine response, what we're going to see is a reduced or a muted response in that we will still have proliferation, just not as much, not as abundant as we had when we had just a little bit of TGF-beta. So while this seems very simple in nature, understand that you can still have quite a lot of fidelity in the way cells respond based on a combination of cytokines. Nothing is going to be mutually independent. Everything works together, that homeostasis that we talked about in human physiology. Here's a paracrine, just an example, the signaling cell, and we have local cells that have the receptors. If you don't have a receptor for a particular cytokine, whether you're local or distant, you are not going to respond to a particular signal. So this is a paracrine. Endocrine would look similar to this. The only intermediate step we would have blood vessels interspersed in between our signaling cell and target, in which would transport those signalers to distant target cells in the body. Now, our classification of glands. First, we're going to start off with this terminology, exocrine. And a lot of times we think of exocrine as the opposite of endocrine. And in some respects it is. Exocrine is going to be different in that we don't rely on blood vessels to transport our cytokine. We're going to produce ducts and have a ductal system that will specifically deliver material to the target. For instance, we have salivary glands. Saliva is going to be a collection of enzymatic products that will degrade material. You do not want those products entering into the bloodstream and degrading your own body. We specifically direct those through ducts into our oral cavity. So those are exocrine types of secretions. Now, with our exocrine glands, we're going to have some principal components that we're going to identify, and most of these are histological in the way they look, but some are also going to be molecular. First, we look at mucus glands. Now, mucus glands, these are the parenchyma, are going to produce mucigens. And when these mucigens are hydrated, when they become water hits them, they're going to become mucin. And mucin is the principal component that you find in this lovely substance we know as mucus. Now, histologically, when we look at mucus glands, here we see a lot of our glandular epithelium, and many of these are going to be rounded in nature because we've cut them in cross-section, but certainly some are going to appear more longitudinal, and we can cut them in that plane of section as well. So you see some of these are going in and out of the plane. A lot of these are going to be nice round cross-sections with a small lumen. Cell borders are pyramidal cells. But in these, when you look at the cytoplasm of the cell, you are going to see a fairly pale look to our mucus glands, and you can see how pale these are. This is going to be some connective tissue. Here we have a duct of the gland, and again, your eye immediately goes to that larger luminal diameter. You're either not going to see the lumen of many of these glands, the parenchyma, or it's going to be quite small. There's a prominent lumen of that parenchyma in that location. So where do we find these lightly stained mucus glands? Well, salivary glands certainly. We're going to have some in the esophagus, stomach. The pyloric glands are going to be in the lower portions of our stomach in the region of the pyloric sphincter. And then Brunner's glands are going to be specialized glands we find in the duodenum. They're producing mucus to neutralize the stomach acid before it enters into our intestines. And you're going to love seeing Brunner's glands because they are a very prominent landmark that will help you distinguish the duodenal portion of the small intestine from the other portions of the small intestine. So mucus, from a histological standpoint, these mucus parenchyma are going to look empty, poorly stained. The cytoplasm is going to be more apical because the nuclei are going to be basally located, very, very intensely staining in that basal compartment of the cell. Now, to contrast mucus, here we're going to talk about serous glands. And again, not necessarily the opposite from a histological standpoint. These are going to stain much more dark than you see with the mucus because principally a lot of our histological stains detect a lot of protein. And that's what we're going to see in serous glands. It's more of an aqueous, watery solution instead of the thicker, more viscous mucus. But these are going to be filled with enzymes, proteins. And when we look for serous, we're going to see those in salivary glands. Notice we also had mucous salivary glands, and we'll be talking about that as we have a mixture of the two different types. In the pancreas, the exocrine portion of the pancreas, we're going to see as serous glands. We also have the endocrine portion of the pancreas, which we're going to call the islets. Gastric cheese cells. These are going to be cells part of our gastric glands, and then Paneth cells are going to be in our intestines. Again, very distinct cells that we're going to be able to identify histologically. Serous cells are also going to be organized into these rounded parenchymal units called acini, but they're going to clearly be distinct from our mucus type of parenchyma because they're going to be much more deeper red, even maybe a more bluish stain to the cytoplasm. The nucleus also is not going to be as basal as we see in the mucus acini. They're going to be more centrally located. And the staining, that deep staining that we see, is because we have so many secretory vesicles filled with these enzymes, and we refer to them as zymogen granules. Zymogen is a term that refers to the content of these granules and vesicles being enzymes. This is a nice illustration because it really shows the comparison and contrast between mucus acini, which is on the right, and serous acini that you see on the left, separated by connective tissue. We have pale staining cytoplasm. Our nuclei are really flattened to the base. These almost look like nuclei of our squamous epithelial cells. And as we move over to our serous, you have nice, rounded, centrally located nuclei, much deeper staining in the cytoplasm, and really very little, if any, of our lumen are visible. That might be the lumen of a serous acinus there, but very little in the way of a lumen being visible. So notice the deeper staining, the rounded nuclei versus the flattened nuclei, the more pale stain, contrasting our mucus versus our serous glandular epithelium. Now, as we saw in our listing, we can have mixed glands as well. And mixed glands doesn't refer to portions of the organ being mucus and portions being serous, like we saw on the previous slide, clearly separated by connective tissue. But we can have mixed glands in the same acinus. And what we're really going to see is that principally an acinus is going to be composed of mucus parenchyma, but on the periphery you're going to have cells that are producing serous material. They're going to be hanging on the outside and injecting through cytoplasmic extensions their product into the middle lumen of the acinus. And that's going to give these serous cells an appearance of somewhat of a crescent shape. And so we refer to these as serous demilunes or half moon because someone early histologically said, you know what, these things look like they're crescent moons or half moon shaped objects. And in this illustration, we can see what is referred to as a mucus acinus, very light pale staining nuclei. There's our basal flattened nuclei, pale cytoplasm. But then you have this crescent-shaped cap on the outside. That's a serous demolune. Here we have another, this is trying to say serous acinus, but I don't buy that because we got some mucus down here as well. Once again, here we have some mucus parenchyma. There you see the lumen of the gland. And here on the periphery, we have this serous demolune. And in this case, you can almost see the point at which the cytoplasmic process is pushing downward toward the lumen to inject its serous material. So there we have a serous demolune. There we have a serous demolune. Not so sure about this. Here we have a mucus acinus. We have some nuclei along the base, maybe another serous demolune there, but you can see most of what we see out here. These are going to be serous acinus, complete serous acinus. But these are mucus acini with our serous demolune. That is our mixed type of gland. Now, we've talked about the different secretory products. We've talked about the histology and the look of these glands. Now we can also classify glands based on their mode of secretion. How do the cells produce these materials? So one mode is called miracrine secretion. This is essentially what we are most familiar with, in that the vesicles containing our secretory product fuse with the plasma membrane, and through a process of exocytosis inject that material out of the cell into the lumen, and that's going to follow the ductal system and take it to its target. The parotid gland is an example of acini that produce their saliva through a miracrine type of secretion. But we can also secrete materials in an apocrine fashion. And in this case, portions of entire cells break off, and portions of the cell, these membrane-bound packages, are actually transported. And so this is going to be an example that we find in our mammary gland, and specifically lactating mammary gland in the production of milk. So apocrine are big chunks of cells. The cells remain. You can see the nuclei stay and these grow back and they continue to produce this material. But this is apocrine. Much more material shed from each cell much more rapidly. Now finally we can get to almost the pinnacle of our modes of secretion. This is holocrine. And when you think of holocrine, I want you to think of the whole thing. And in this case, an entire cell dies and the entire cell becomes a secretory product. And the sebaceous glands that you have in your skin associated with your hair follicles is an example of how we produce this sebum, this material that makes your hair more supple. That is going to be a place where we have an example of holocrine type of secretion. We can see other cells that are being replaced as cells are lost. So these glands don't simply disintegrate and go away and you never make more. But as cells are lost, other cells are undergoing mitotic divisions to replace and maintain the integrity of the gland, yet still produce the secretory product. Now we get to the simplest of our glands. These are unicellular glands, and so they're not really what we think of as far as an organismal unit or an organ unit, tissue. These are individual cells. They're going to be placed in different places. Our alimentary canal, we see them largely in our respiratory tracts. These cells are producing mucus and they secrete it at the surface. They will have attachments to the basal surface at the basement membrane, and they will have extensions that reach all the way to the lumen of the particular organ within which they reside. Here we can see some goblet cells, and they're goblet because the mucus is assembled into this large unit that is more apical in nature, and then the cytoplasm is confined much lower down, and it forms sort of the neck or the base of what we think is a goblet or a wine glass. You might think of it like that. And here you can see the secretory product coming out at the free surface or the apical surface in this particular area. Here we have some examples of sections through colon in which most of what you're seeing, the epithelium-wise, are going to be goblet cells. And in this diagram we can see we have a basal nucleus and these large mucigen granules, and you can see they're secreted at this apical surface. That is a diagram of cross-section of a goblet cell, a unicellular gland. So we've talked about exocrine glands emptying into a ductal system. We've talked about the different modes in which these secretions are produced. Now we're going to classify these so we can identify them histologically. And one way that we can subclassify exocrine glands is by the shape of the secretory units. They're either going to be tubes, tubular, rounded structures much like berries, which we call acinus, or they might be extended tubes at which the very end is going to be rounded in nature, which we can refer to those as tubulo-acinar. Now, you can use the terms acinus or alveolus, but I really prefer that term acinus because alveoli, I like to reserve that for our discussion of respiratory epithelium and respiratory function. So stick with acinus. Latin means grape. It's just a small ball. And really, when we look at those, and that's what that reminds me of, a grape. So we're going to stick with acinus as we talk about our definition and classification of our exocrine glands. And here we see an example. The blue is going to be the ductal system. And then this rounded parenchymal part with a lumen in the center, that is our acinus. Now, you're not going to see clear boundaries like this. These are white lines that have been produced by this particular image and the person that posted this, but it gives you an example of the nice rounded nature of this parenchyma and the open lumen there in the middle. Once again, as we've mentioned before, we have these acinar cells, but they are going to be pyramidal in nature. So they're going to be much broader along the basal surface, and this is where they're attached to the basement membrane. The apical surface is where they empty into the lumen, and they're going to be very restricted. That's going to be the pinnacle, the point of our pyramid at the apical surface. And then we're going to have those lateral surfaces, the junctional complexes, in which we are going to form the adhesions and attachments between adjacent cells within that same acinus. Now, we've already seen different sections, and we mostly see the round cross-sectional views of those, but we may also see elongated sections, very long pieces. But principally, when we look at these glandular epithelial cells, they're going to look like the Trivial Pursuit piece, rounded, in which you have the little pie pieces, and those, you can think of those as the pyramidal cells. That's our typical look for our acinus type of exocrine gland. Tubules, as you can see, you have the ductal system in blue, and these are much more elongated in nature. And with these, we're going to find these more common for mucus types of glands. So esophageal glands, pyloric glands in the stomach, Brunner's glands, and the duodenum and salivary glands are going to be tubular in nature, producing mucus. But we're also going to find these in sweat glands and the gastric glands in your stomach. They will be tubular in nature. And when they're tubular, you're going to see many more of these cut in longitudinal section rather than just the rounded acinar cross sections. So as we look at a histological section, you really have to take a survey of the field in which you see. If all of the parenchyma are acinar in nature, then you have to understand the random position that they were in when they were cut means that all of the parenchyma were acinar to begin with. However, as you survey the field, if you see some rounded cross-sectional cuts and you see some oval shapes, some longitudinal, you're probably looking at mostly tubular glands in which some of those have been cut in cross-section. So plane of section is going to be something we'll talk about quite a bit, especially in lab, as we look at our different divisions and classifications in different tissues to help us understand really what we're looking at. Now, cords are a little bit different. You may think of these as almost like tubules, but I want you to think more of them as partitions and walls as opposed to twisted up pieces of spaghetti that are piled up. These cords are principally going to be found, and one of the places we find them certainly is in the liver, where we'll see these hepatic cords very, very prominently. But we're also going to see that these are mostly going to be not for exocrine so much, even though we do have some of these, but more endocrine in nature, and we're going to rely on the bloodstream circulatory system to transport these where we need them to go. But cords, As we mentioned there at the bottom, we've got the liver. We're going to see these cords are also in the pancreatic islets, that endocrine portion of the pancreas, the parathyroid gland, the adrenal cortex. Some of these layers are going to have cords or plates that we're going to see, and we'll identify them as we get into our organ histology in the next several units throughout the semester. So we've classified our exocrine glands, and even to a slight degree our endocrine glands, based on shape of those secretory units, tubular, acinar, tubuloacinar, even corded in nature. But now we're also going to organize them based on the secretory unit. Is it a single tube or acinus, or is it multiple? And then we can also describe and classify this gland based on the organization of the ducts. Is it a single duct, or is it multiple in nature? So here we have tubular, acinar, tubuloacinar. That's what we talked about. But when we get to having multiple units coming off of one duct, then we're going to refer to that as branched. Now this is where it gets a little tricky. If we had only a single unit, we would simply call this a simple tubular, simple branched, but simple acinar. But since you can see our parenchymal units in green, we have multiple units. This is tubular. Here we have acinar. And I'd almost describe this as tubuloacinar. Maybe that needs to be a little more extensive before we do that. But we have these three units on a single duct. So we're going to refer to this as simple because we have one duct, branch because we have multiple secretory units, tubular gland versus an acinar gland. So here's our simple tubular, here's our simple acinar. Now, if in fact the ductal system has multiple units to it, then this is going to be referred to as compound. And in this case, they have drawn this illustration so that each one of our parenchymal units has its own ductal system emptying into first this duct, and this duct. And we'll see a lot of these in our exocrine glands. So if the ductal system is branched, or there's multiple ductal systems, I want to avoid that word branched, then we refer to that as compound. And then we will add the shape of the parenchymal unit. Compound tubular, compound acinar, compound tubuloacinar. And I think you can appreciate the subtle difference between tubuloacinar and acinar. We're not going to get into and split those hairs. Now, it is likely that you could have multiple ducts with multiple parenchymal units in which you would have a compound branched tubular or acinar or tubuloacinar duct. But again, we are not going to get into that level of looking at our glandular epithelium to get to that point. But those do exist as well.